Hello, in this video I'll explain the negative inducible control of the lac operon as well as how mutations within the lac operon can impact these controls. To begin with, the lac operon is an important operon within prokaryotes and has the purpose of allowing the cell to turn lactose into a source of energy. For the purposes of this explanation, I'll focus on 5 regions of the lac operon, with these regions being lac I, which is the regulatory gene that encodes the repressor protein for the lac operon, the lac promoter where RNA polymerase binds, the lac operator where the lac I repressor can bind to and block RNA polymerase, lac Z, which is the gene that encodes beta-galactosidase, and lac Y, which encodes permease. In the absence of lactose, the lac operon is repressed. This can be seen in the diagram as the repressor protein is transcribed and translated from lac I. This repressor protein has, for simplicity's sake, 2 binding regions, one region that binds the lac operator and one region that binds allolactose, a derivative of lactose that turns off the repressor. In the absence of lactose, the allolactose is not present to bind the repressor, allowing the repressor to bind the operator. This prevents RNA polymerase from transcribing the genes for beta-galactosidase and permease. The repressor blocking transcription is negative control. In the presence of lactose, the lac operon is induced. When lactose is present, its derivative allolactose binds to the repressor, changing its shape so that it can bind the operator. This allows RNA polymerase to transcribe lac Z and lac Y, which eventually leads to beta-galactosidase and permease. Allolactose inhibiting the repressor, allowing for transcription to occur, is inducible control. I will now cover an example of a partial diploid lac operon that has several mutants. The top strand has a mutation for a super repressor that cannot bind allolactose and be turned off, as well as a defective promoter that won't bind RNA polymerase. The bottom strand has a mutation for a repressor that cannot bind the operator, a constitutive operator that cannot bind to the repressor, and the lac Z that encodes a defective beta-galactosidase. In the absence of lactose, the superrepressor that is transcribed and translated binds to the normal operator. However, it cannot bind the constitutive operator. The defective repressor cannot bind either operators. The RNA polymerase fails to bind to the defective promoter on the strand with the attached repressor. However, it does bind the promoter on the bottom strand and transcribes the genes that translate for defective beta-galactosidase and permease. Therefore, only wild-type permease is produced in the absence of lactose. This is also true when lactose is present, with only wild-type permease being produced. This is due to the translated repressor proteins being either defective or not being able to bind allolactose, resulting in no difference from when lactose was absent.